Welcome to Moonshot Radio, where we stop and take a moment to reflect on the rapid pace of technological change. We meet the people reprogramming our future to explore what's coming, how it's reshaping our world, and what that means for our human experience. I'm Linda Du, a fintech founder, investor, and adventurer, posing the question of what it means to be human and exist in society in the age of technology. Our guest, Heather Dewey-Hagborg, is an artist and biohacker known for reconstructing human faces from discarded DNA. She shines a light on the shadowy world of biosurveillance and the developments in biotechnology that are shaping our future out of sight. She raises urgent questions about privacy, identity, and control. Heather's projects reveal how much of ourselves we leave behind and how our DNA can be used by governments and corporations to define our story. In this episode, we talk about genetic data, forensic art, and the unsettling beauty of our biological trace. So a lot of your work focuses around these themes that are both inherently humans, such as biology and DNA, but also relate to technology and society as a whole. I saw that one of the key themes you explore is biosurveillance. So could you talk to me a bit about that and some of the projects that you've done to explore that space?
Biosurveillance is basically looking at how biology is used to track us, monitor us, and analyze us. So looking at things like DNA, microbes, looking at things like face recognition, fingerprints, and then also things like perspiration, respiration, the giving off of heat of the body. So it's all of these aspects that are just inherent to having a physical body, being in space, things that we can't control or have much agency over, that can be used to follow us, to track us, to turn us into data, and then used for whatever kind of purposes, whether those be governmental or corporate. So that's the kind of big picture around biological surveillance, is thinking about how our bodies are turned into data, essentially. The first project where I really learned that biological surveillance was happening was this project, Stranger Visions. This started in a therapy session, actually. I was sitting in a therapy session, staring at this print across from me on the wall. The glass covering the print was cracked, and there was this hair stuck in the crack. This was 2012. I sat there staring at this hair and thinking about the person who'd left it and wondering about them and thinking, given all of the technology that we have available today in 2012, what could I learn about a person from a hair? And that launched me down this path that has taken me to where we are today, but that has taken me through this world of biological surveillance, through community biology labs, learning how to extract DNA, learning how to analyze it, learning how to predict things about people from DNA, and learning a lot about the limitations of that and the subjectivity involved in those processes as well. And so that first project, thinking about that hair and what can I learn about a stranger from a hair, that was Stranger Visions. In that project, I picked up these things like hairs and cigarette butts, chewed up gum, nail clippings from the streets and public bathrooms and waiting rooms of New York City. I took those things to GenSpace, which was the world's first community biology lab at that time. Community biology lab means anyone can come in and learn the basics of molecular biology and learn how to do experiments themselves. I took the cigarette butts in and over the course of months learned how to extract DNA, amplify it, and start to analyze different genes or genetic variants related to appearance. And then I took those into a computer model and used that to manipulate that model in different ways to generate possible faces based on that data. So the long and short of it is extracting DNA from found items on the street and generating 3D portraits based on that information. I exhibited those portraits along with the artifact they were generated from and the information I'd extrapolated from the data and the metadata around where I'd found it and when.
And so the work that you did sounds almost innocent in a way because it's almost an exploration of what could someone look like. But then there becomes this other question, which is, okay, when we turn DNA into data and use these large data sets, and then we start to do the categorization, and then we start doing some labeling and making judgments and decisions about this, where it actually becomes quite dangerous. So I'd be interested to hear your thought about what is the turning point at which this becomes something less innocent, and what have you learned about society and judgment throughout this process?
Thanks so much for asking that, because it is such an important part of my thinking in that project already, but in particular, how my thinking unfolded afterward. So in that particular project, so starting that journey, as you say, it was a little bit innocent. So it was a little bit of a research project, starting with this question: how much can I learn about a stranger from a hair? That leads me forward. And it leads me to learning that there is research happening to try to make these predictions about strangers, but really about forensic DNA evidence, right? So I'm finding papers in 2012, I'm finding these papers from scientists trying to predict basic traits from forensic evidence, DNA evidence. I could see that what they called forensic DNA phenotyping was coming, and that this would be used in criminal investigation context. And so one aspect of that project was to show people that this new technology was just on the horizon. I thought it was still 5 years away in 2012. It turned out it was closer than that. And kind of warn them that this is something that's happening. We should start thinking about it and start kind of thinking about what it means. In that project in particular, I was very focused on the privacy surveillance angle, saying we are shedding this information all the time. We have no control over it. Nobody's talking about this. Let's start thinking about what it means. DNA is increasingly legible. It's cheaper and easier to read than ever. What does that mean? So that was really the focus of that piece. A couple years later, this company, Parabon Nanolabs, launched a service they started selling to police, claiming to predict mugshots from DNA evidence. This was very interesting to me, but very alarming, because I'd just gone through this extensive research project. I'd read everything that there was to read about what you can predict about someone from DNA alone. And I knew in my own process how subjective that act had been, how I had generated 5, 7 different possible portraits from one DNA sample, and then used my artistic subjectivity and said, okay, this face makes me feel something, or this face reminds me of someone I saw once, and gravitate towards those and choose those as an act of artistic subjectivity. But what would the criteria for the police be in making these decisions? Where is their subjectivity going to enter into that process? What kind of transparency would there be around that? As we know, there wouldn't be any. There wouldn't be any kind of public transparency around those decisions. It would be presented as some kind of authoritative image, and there would not be a discussion around the limitations of that knowledge. I was concerned already in that moment that those images could be used as another kind of racial profiling, basically as a stereotype. And those first images that were put out by that company, if you scroll through them, you see that they had very generic portraits. They were putting out these prototypical different faces and making tiny changes based on the very limited genetic data they actually got. So in that moment, I started trying to think, how can I invite people into thinking more critically about this technology also? So Stranger Visions was very good at getting people riled up and thinking about surveillance and privacy issues. But how can I get people thinking also critically about the construction of these images and what the political implications might be of generating them? And that led me down this next branch of my artistic path, which was working with Chelsea Manning. So Chelsea Manning was a whistleblower who was arrested for the information she made public, exposing the prevalence of civilian deaths and torture in Afghanistan and Iraq wars, among many things that were in the documents she revealed. She was sentenced to 35 years in prison, and at her sentencing, announced her gender transition. Chelsea was this kind of hero whistleblower figure within the community that I was in, which was, I guess you could say, this kind of art and technology community, but also left community. People, we were really inspired by what she had done in exposing the brutality of these wars that the U.S. had been waging for so long already. So long story short, Paper Magazine is doing an interview with Chelsea Manning through the mail. Nobody can visit her. Nobody can photograph her. She's been completely cut off from public view. You could say that her image had been suppressed, held away from the public since that moment of her gender transition. So they contacted me and said, we're doing this interview with Chelsea through the mail. Would you be interested in doing a DNA portrait? And it turned out Chelsea had also seen Stranger Visions and thought it would be a cool idea to try something like that. She sent me Q-tips that she swabbed her cheeks with from prison. And I went through that same process as with Stranger Visions. But this time, the idea was to invite multiplicity, but also use the technology for a different purpose. Use that technology for something that could be liberatory instead of just oppressive. To give someone whose face had been erased from the public, to give them that public image. And so we ended up working together on multiple projects. On the first project called Radical Love, it was these two faces, two portraits. One was parameterized gender neutral, you could say, or the sex parameter would be set at a neutral point. The other one is parameterized female. That first gesture was really building on these ideas that we're not limited to our biological sex, but also trying to pull out and show people that these computational models, these AI models, facial recognition models, are very limiting in terms of how we think about things like sex and gender. The whole idea that you could represent as if you would have a slider between male and female within a facial model would be absurdly limiting. The two faces are shown side by side, and the viewer can walk up and look at them and think about that, think about what they take away from the differences between these two faces, and hopefully critically reflect on the limitations of these computational models. And that was something that we were both very interested in because Chelsea Manning also had a background in doing information analysis and was very much into computer science and AI as well. And I'd spent the decade before basically doing art, engaging with AI.
I read about that and I thought it was so powerful in that there's so many layers of data, both from a biological perspective, but even from the national intelligence perspective, but also in the identity. So I'd love to hear about what was the impact of that work on Chelsea, either through that work or through your other works. How do you explore the interplay between determinism and agency when it comes to identity?
Oh, yeah. That's great, too. So the story with Chelsea goes on. That work premiered at the World Economic Forum, which was a surprising place for it to premiere. But it was an amazing place for it to premiere because you think this person has been thrown in prison. The key's been thrown away. Nobody is really paying that much attention anymore. And her face has been suppressed. And then we're taking her image and putting it right there at one of these most elite political events of the year. You have world leaders congregating there, and they are confronted with her face. So that felt like a tremendously meaningful place to actually intervene and put her face there. And as it continued, so as Obama was leaving office, there was a campaign to commute Chelsea's sentence, to basically say she'd already served X number of years. I'm trying to remember how many it was exactly, but it was quite a few already. And to say she's already served these years, that's enough. And that's the idea of commuting her sentence. So there was a campaign to commute her sentence before Obama would leave office. And there was a lot of activism around that. And I wanted to also work with her on something to try to campaign for clemency. And what we came up with was to work together on basically a comic book. So we wrote this comic together that narrated the story of us collaborating on these DNA portraits, and it walks through those steps, like I narrated to you, sending the cheek swabs, generating these faces, and what some of the limitations are, get people thinking about that. But then the main thing was that we ended with this speculative last page. So the last frame of the comic book, we picture at the top, it says that Obama commutes her sentence and Chelsea walks free, and she walks into a gallery and can see these portraits in person for the first time. Because at that point, we'd shown these portraits in different exhibitions around the world, but of course, she's never actually had the chance to see them other than as just a small print that could come to her in the mail. And we put that out, and then the amazing thing was that it came true. Obama commuted her sentence. And a few months later, she did walk free. And we had an exhibition together, and she could walk in and she could see those portraits in person for the first time. And this was really one of the most moving and meaningful moments in my life, actually, but definitely in my artistic or professional life. And for that exhibition, we had this gallery to ourselves, 1,000 square feet. And we started to think, what should we do with that space? We just have these two portraits. How do we want to scale that up? What else can we do? And we had the idea to take this way of pushing against the limits of the algorithmic model and to take that further. So to say, yes, we've shown you 2 different possible Chelseas, but how about 30? Let's fill the gallery with possible Chelseas, all generated from the same DNA, and use that as a way to show that it's too limiting to just put this one face on a DNA sample and say, this is the suspect. Rather, you should have this multitude of probabilities, of possibilities that you could predict from the same DNA sample, and show how many different stories you can tell using that data. So probably Chelsea did that. We filled the gallery with these 30 different portraits. Each one represents a different kind of parameterization based on the same DNA data, looking at different probabilities or possibilities in that data. They're arranged at a range of different human heights. So when you walk in, you can enter into this crowd, and it's like a crowd, the crowd that stood with Chelsea when they were campaigning during the activism for her release from prison. It's like this crowd is standing there with you, and it's all of these different possibilities of her DNA. That was one way of trying to kind of blow that up and explode our idea that DNA would be this simplistic kind of reduction. And so to the question about determinism and reductionism, it's a little tricky because it's definitely the case that we are not just our genes. No scientist would say that. It's clear that there have been a good handful of single gene diseases that have been identified that has led to different kind of medical treatments, which is great, but it's so limited. Those number of single gene diseases is so small, and it turns out that most diseases are so much more complex. And that's just talking about disease. When we're talking about what makes us who we are, not just what we look like, but what we act like, it becomes more complex. It's clear that this is not just a question of genetic determinism. Most scientists that I've talked to would say, for example, that you're 1/3 your genes, 1/3 your environment, 1/3 this epigenetic intermingling of these 2 other factors, which is a simplistic way of thinking about it. But it is kind of interesting because if you think about these different thirds, there's still a determinism there. It's just not a genetic determinism. So I think it's too simple to say that we are just our genes. But to say that we are almost entirely determined by the confluence of our genes and the social factors is hard to argue against.
A couple of themes you touched upon that I'd love to explore more with you. One is about idealism, where in this comic book you created a scenario that would have been ideal for Chelsea and it actually got realized. And then the other edge of that is perfectionism. One of the big critiques of genetic engineering is this idea of designer babies, and people being able to pick other genetic aspects that are essentially a reflection of societal constructs, be taller, have a certain eye color. So I'd be interested to hear what is your take on this genetic engineering and the fact that we can use determinism. Hypothetically, we might have the tools to create perfect versions of ourselves.
So I think there's this quest towards perfection, as you put it, this quest towards ever improving, making people better. And this can be a very devastating impulse. There's no question. The major concern here is that we lose our diversity, that we lose our individuality when we try to engineer the human species in a way that optimizes it. We've seen this before in many different places in the world. Many people are familiar with Nazi Germany and what happened with concentration camps and eugenics in Nazi Germany. But there was a huge eugenics campaign also here in the United States and in many countries around the world. Some countries really up until very recently, like the very late 20th century. So this impulse towards eugenics, which involves things like sterilizing people so that they can't reproduce, this has been a recurring theme in reproductive politics for 100 years or more. That impulse seems very strong. So I bring up that history because that feels like it's been a strong and consistent impulse in society. I don't think that we can just wave that away now and say, we've changed, we've come so far, we won't go back to doing things like that. I think it's really worth thinking, how do new technologies enable these kinds of eugenic practices? So this is not just fear-mongering, I think, to think those things through and think them through in depth and detail and wonder about it. And I felt that way for a long time, but I feel much more concerned about it recently, given the political orientation that is happening in the United States, but happening around the world, which is a progressively increasing xenophobia, fear of those who are different. If you think about how these impulses are growing and increasingly shaping politics, pushing against migration, things like that, that makes me very worried about how biotechnologies could be used to serve that agenda. So I'm concerned about things like profiling migrants. I'm concerned about things like profiling the population. Maybe it's already started actually. People applying for asylum, for example, can have their DNA extracted and analyzed to try to tell if they're telling the truth about their story. So that is something that happens. It's not always happening, but it does happen. And I'm concerned about the trajectory of these kinds of impulses. What happens when we start looking at the general population? If you think about the MAGA movement here and the way in which people feel emboldened to express white supremacy, for example, xenophobic rhetoric, what happens then when you start analyzing people's DNA? If you look at DNA, you can tell so many different stories. It's like we learned that already with Snowden, how data can tell so many different stories about you, and they're not necessarily true. You can spin data in different ways to serve your agenda. DNA data is no exception to that. You can spin different narratives. You can look at our DNA and say, well, look, we're almost all the same, 99.9% the same. Or you can look at it and say, but we're 0.1% different. That's the part that really matters. So that data can tell different kinds of stories and serve different political agendas. And so my concern is when people start looking into those tiny differences and trying to tell stories about purity through looking at DNA and biological data. That's very much on my mind these days. And it hasn't manifested in a project yet. I'm still in this kind of research and mulling it over phase, but it feels very important and urgent to me to find ways to address this and also try to expose the places where this is already happening and where those trajectories are starting to unfold. Does that make sense, this kind of trajectory that I'm laying out?
Yeah, and a follow-up to that is America is very much, if you look at global stock markets, 80% of the global stock markets are based in the US, either New York or San Francisco, but it's also very much leading the world in terms of private technology development.
Yeah.
And I'd love to hear your thoughts about some of these trends and how they've changed. I think about 10 years ago, big data was all the rage, and already there was this identification that data itself was biased in the way that it was collected, but also in the way that it was applied. And now we've got this big paradigm shift of artificial intelligence, which also comes with its own dangers. So I'd love to hear how you think about how these big social shifts are also combining with the technological shifts.
Yeah, absolutely. And the big question really for me is how biotechnology will line up in relation to those other waves that you're talking about. If we think about what happened with computers and the internet and then data and social media and now this AI wave and the different impacts these tools have had. And it's funny, actually, because I'm just finishing up on editing a book about the early internet. So it's like an oral history of the early internet. We talk to activists, scholars, and a lot of artists about what they were excited about in those days. Also, what they were already critical of, but what they were excited about because it's gotten so lost, it's hard to imagine now. We've gone so far in this destructive direction that for so many people who weren't living through that moment, they can look at that and say, how could anyone think this could ever have been a good idea? Working on that book along with York Bloomtritt, we're editing this book together to try to narrate in people's own words what they were drawn to in those moments. So then thinking kind of what comes next? Where does biotechnology take us? What are the impacts going to be? What's the potential? How much agency do we have right now to change that? This is the most pressing and important thing from my perspective. Where do we still have agency to make a different future, to do something with the next technological wave that could be not destructive, that could make us more humane, that could make us connected in a way that is generous and considerate and kind? It might sound naive or too hopeful, but I can't see why. If technology can bring out the worst in us, why can't it bring out the best? I can't see any reason why not, other than that there's a lot of social forces and economic forces working in the opposite direction. But all of those things to me seem like things that are up for debate, up for pushing in a different direction. As overwhelmed and sad as I get very often looking at the trajectory of history and technology and the impacts that have happened over the last decades, I still have some kernel of hope that we could move forward. Maybe not with AI. It might be too late with AI. I'm not sure, because we can get more into that, but there might still be some potential to shape that in a positive direction. But definitely with biotechnology, because, well, there's a lot of bad things that could come through these genetic technologies. Most of the things that have actually happened today have been for the social good. I would say on the whole, most of the things have been life-saving technologies, life-improving technologies in a medical sense, medicines, things like that. Those have been real, genuine, positive things that have come out of biotechnological research. It's another conversation about whether they're distributed evenly enough and who has access to them. That's another very important aspect. Generally, I think that the development of the technology has been a net positive so far. There's still room to shape the forces that are going to come from biotechnology into something that could have this real positive impact on the world, that isn't going to be just furthering our most destructive impulses. As much as there's the potential that people can look for narratives of purity in genetics, they can also look at it and say, wow, we're so intermingled with each other. We're so multiple. We've been moving around this planet since the beginning. We're all full of so much diversity and such a multitude of difference. That's just as true if you were to talk to scientists about it. I think that there is still a lot of room for hope and potential, but we have to act fast.
One of your works that I was looking at beforehand was Xeno in Vivo, which I found fascinating because it's almost the opposite of purity that you spoke about. What was the concept for that? Where did it come from? And what was some of the feedback that you got?
Yeah, so that project was a little bit of an outlier in my artistic trajectory in the sense that so much of my work is about humans, genetic profiling of humans, let's say. And that project, I was really thinking about our very close relatives, which are pigs, and researching into these technologies of xenotransplantation. So looking at how we're engineering other species, but in particular, engineering pigs to be more like humans so that we can use their organs for organ transplantation. And this was a technology that when I first heard about it, my initial impulse was, oh, that's really problematic that we engineer this whole species just to kill them and use their organs. Going through the process of that research, I would say I still feel that way to an extent, but I definitely came out with a bit more nuance and a little bit more reflection on the complexities and the possibilities of that technology. So it's not the piece Xeno in Vivo, so it's an opera about xenotransplantation. It's an opera that is constructed, the libretto, the words of the opera are from interviews that I did with scientists, archaeologists, all these different people who work with pigs, genetically engineering pigs, but also looking at this long history of how humans have shaped the pig genome through selective breeding and domestication over the millennia. It's this long look at this relationship between the human and the pig, or the human and the domesticated animal, you could say, really thinking about this. What's often posed, scientists often say about genetic engineering, scientists who perform genetic engineering often say, we're not doing anything new. This is just a continuation of millennia of manipulating genomes through breeding. And CRISPR, it's just the latest step in this trajectory. And going into Xeno in Vivo, I really wanted to probe that question. That was my personal interest in it, saying, okay, I've heard this narrative a lot from scientists. Let's dig into that and hear different perspectives and make some kind of piece out of that that can let audiences reflect on that themselves. Because I think it is more complicated than that. I do think it's not totally wrong. It's true that we have been manipulating the genomes of animals for millennia. And it's true that we've created new species using very low-tech methods of breeding. We've created many new species, a lot of the plants and animals that we are familiar with today in that way. And it's also true that CRISPR gene editing blows open the door and creates so many new potentials in terms of how quickly you can make new species, in terms of the kinds of species that you can make, the genes that you can cross from species that would never breed with each other, for example. So there is also something radically new happening with these CRISPR gene editing technologies and genetic engineering generally. And so the Xeno in Vivo opera is using the words of these scientists and archaeologists to bring the audience into thinking about these relationships between the human and the animal.
Beyond the biological or genetic design component, something else that I was thinking about goes back to when you mentioned that in our DNA we are very, very much similar, and that applies to pigs as well. Yet there's almost this facet of human identity where we have to consistently prove to ourselves that we're better than other animals. And so I wonder if that idea of human existentialism came up during that piece or any of the other works that you've worked on. What is it that actually makes us human?
Yeah, definitely. In seeing ourselves reflected in another species. In looking at the xenotransplantation research, there's 3 countries that are the main countries doing this research. That's the US, Germany, and China. And these are also the main pork-eating countries in the world. It's interesting, right? Because it indicates that there is a willingness to kill these animals, even though they're incredibly close to us. And at the same time, you have to think, but you see how close we are. I mean, if you are one of these researchers, you're spending time there with these animals. You're seeing how much personality they have and how developed they are in their ability to think. And the scientists, when I met them and spent time in those facilities, it's absolutely true. One of the scientists I met with who does this research is George Church. He said, I'm a vegan. I thought that is so interesting. What an interesting juxtaposition to be vegan and also working on genetically engineering animals to provide organs for humans, right? And it feels like a contradiction at first. And for him, he unpacks that in saying that it is a bit utilitarian, let's say, that it's the good that can come out of engineering one animal to provide multiple organs. Ultimately, we're talking about a small number, and it's going to have this really net positive impact and so on. So it's a kind of utilitarian calculation. But it is definitely walking around this territory and this question and asking people to think about that and how they relate themselves to these animals. Many people in the audience watching the piece do eat pork. And a number of people come out and say to me, I didn't never want to eat pork again after watching this opera. So I feel like, well, that's something. One of the pigs in the opera, it's kind of documentary in the sense that I'm talking with these scientists, but it's also speculative. So there's these speculative pigs that also appear in the film part. And one of them has a human nose. It's very much thinking, what if we made it visible? This similarity. When you engineer a pig to be more like a human, you can't really see it. The pig basically looks the same, even if their organs are changed to be like a human's organs. And so I thought, what if we engineer also the pig's face so that they look more like us? How would we feel then about taking the organ? I hope that people just think about it. It's not so far down the line that they might be confronted with that decision. You know, some percentage of us will be needing an organ at some point. And to consider, would you take an organ from a pig? And if you did take that organ, how would you relate to that animal? How would you honor that animal that made a sacrifice for you that it couldn't choose? Unlike the human that donates an organ, the pig has no ability to consent to that gift. So I hope that the opera brings people into thinking through those questions.
When you speak, it reminds me of a book that I've read called Tender as the Flesh. It's by an Argentinian author. And I'm not spoiling anything. It's in the first chapter, but the whole concept is that there's been some flesh-eating disease that's killed out the animal population, but people are so hungry for meat that they start breeding a subclass of human that they use for eating the meat. And so maybe if I just use that to segue into ethics and society, how does biotechnology impact equity within society? I think the most obvious example would be like who has access to genetic engineering treatments, but I'm sure within your work there's many other facets that you've also looked into.
So we've talked about the impulses towards perfection, towards modifying ourselves to try to optimize. These are big aspects of it. And then we've talked also about the surveillance aspect of things. So this is another thing that I would stress there, that there is this way in which biotechnologies, they accelerate this kind of population thinking. It's not that it has like a specific positive or negative thing, but it is just, it's a way of structuring how we think about society and humanity. And it leads to a certain kind of way of quantifying. You know, so suddenly you can enumerate in data and quantify these different aspects and attributes of populations. And so for a long time, early biology was being used in just a descriptive way to think about how do we create these categories to separate people out. So this is definitely one of those big ethical questions around categorization and how DNA technologies and the newer technologies that are coming after DNA technologies are much more about looking at expression, microbes, or viruses. There's so many of these technologies that are so fascinating, where we can learn so many things about the entities that are in us. But of course, they can also be used to put us in some kind of box that can have its pluses and its minuses. It can be useful for medicine, but it can also be a way of segmenting people into different groups that can lead to different kinds of antagonism or discrimination.
Something that I am interested in is longevity and this idea of legacy. I spoke to another biologist a couple of months ago, and something he was talking about was cloning. But then also I spoke to someone who does cryonics, where it's not the same as cloning, but you actually extend your own life. And I'd be really interested to hear about your perspective on trying to live forever through biology.
I can't say that I've done much research into that or too much critical thinking about the longevity movements. It generally feels, on the one hand, like something that is intuitive, right? We all would like to live healthier, longer lives. That's pretty fundamental. The question is, what will that mean in a system unless things have radically changed in a system where things are so unequally distributed? I think that is the main critical question in regard to those longevity technologies. I'd be curious also if there's things that are coming up for you when you're thinking about it, what your concerns might be. But that's the big one that seems to hit me. If you have a 1% or less than that suddenly is living for a much longer time, or cloning themselves and living into multiple generations in the future, what does that mean for the rest of us that aren't able to do that? I mean, do we end up in some kind of feudal arrangement where you have these elite beings who end up being all powerful because they have these special abilities? But I don't know. What are you thinking with longevity? I'd be curious what's on your mind with it.
Yeah, so my family comes from China, and we were very used to structures, and it's almost the interface between social structure as well as individuals linked to that. So, the very famous cult of personalities that many dictators have had. But what I found quite interesting when I was pondering this question for myself was, if you had a dictator in power, they could live forever, but often when they leave, the structure around them is allowed to change. But then it's like, because you have this one person in place and they have created this whole structure around them, that also serves to oppress a population. I was thinking about it as like, what is actually oppressing us? Is it the structure or the person, or the fact that the person and the structure are very deeply intertwined? So it's also quite abstract, but it's about a lot.
It's a really great question and a kind of speculative scenario to think through. And certainly in this moment, one can only think of a few people that might be wishing to live forever, and it's a terrifying thought.
And even just from a biological perspective, because it does somewhat reduce genetic diversity, especially if you combine that if you do clones, you could basically seed out a bunch of clones of one person, and so then that naturally reduces a bit of genetic diversity. But then if you also combine that with another portion of the population doing this genetic engineering to have these designer babies, it's what does that mean in terms of perfection?
And it just feels to me like it's always kind of disappointing, because it just feels like the wrong direction for our energy. We put so much energy into appearing like these great people, and it's so narrow and individualistic and tied in with our egos that we look as good as possible, and that we project some idea that we have this great life. We're living our best lives all the time on social media or whatever. And it feels like it's really the wrong direction. Like, we really need to be reorienting towards the social, and thinking about, what does the public need? Like, what do other people need? It's not just about me living forever. How do we lift people up so the majority are living longer? This would be the antithesis to the Elon Musk approach or Peter Thiel, wanting to live forever, is like, well, how about making life expectancy, even just in the US, longer for the general population? That seems like the important goal. That's like the socially minded goal that I would wish that we could be oriented towards.
And then biobanking.
Yes, exactly. I knew I was missing one. I worked on this installation recently at the Cooper Hewitt Museum, which is in New York City, up by Central Park. They had this triennial exhibition on the idea of home. They invited me. When I heard home, I just started thinking about where our cells and data and DNA lives. Like, where's the home for our bodies, for our fluids, for our data, for our cells? It lives in these places called biobanks. These are facilities that have deep freezers, that have vats of liquid nitrogen, or sometimes just chilly closets, and they store blood and tissue and DNA and different kinds of cells in these deep, cold facilities. And I got interested in these years ago when I was working on a film called T3511, which tells this story. It's kind of like a sci-fi stalker romance story about a biohacker that falls in love with an anonymous saliva donor. So she buys the saliva online, sends it to 23andMe, starts learning more and more about this person through their DNA, traverses different DNA social networks to basically track them down, finds them on Facebook, figures out where they live, goes through the whole genetic privacy nightmare scenario. It's told in a way that is intermingled with this idea of falling in love with someone and exploring what it means to fall in love with someone as you are like piecing together the data about who they are. So that was T3511. Researching that film, I got interested in biobanks and thinking about what's happening with our blood. When you have a blood draw for a medical purpose, where does that go then next? And I wanted to know the answer to those questions. And what's happening with the DNA data from all of these tests and biopsies? Where does that go then? And that led me to realizing that when we are born, in most places in the world, this is true, newborns have blood drawn and it's analyzed for different kinds of conditions, conditions in particular that can be treated right away and have beneficial health effects. But the blood, that is kept in a biobank. And so that blood can be stored for decades or indefinitely, depending on the country or the municipality. And that's just one of so many of these biological samples. So there's these newborn bloods, they're called blood spots, where they prick the heel of the baby and put it on a card of paper. Those are stored in a multitude of different ways. Thinking about the United States, each state has different guidelines and regulations around how long and where and how protected they are, whether they can be used for research or not, whether the police can access them or not. And that's something that everyone born basically can't opt out of. And you wouldn't want to, because the life-saving benefit that comes from that is so important. I mean, you wouldn't want to opt out of the newborn screening program. But there's very little discussion about what's happening with the things next. Where does that live on? Who's accessing that data and that information? And then you realize that that's just one little bit of the story. And that all of the fluids and cells and all of the detritus that can come from our medicalized lives can have these afterlives. The first instance of this was the story of Henrietta Lacks that you might be familiar with, that was famously told by Rebecca Skloot in The Immortal Life of Henrietta Lacks. It tells the story of an African-American woman in the 1950s who has a biopsy taken from her cervical cancer that is transformed into the first ever immortalized cell line. The first cells that could be propagated over and over again for medical research. That story rightfully generated a lot of interest and a lot of outrage and concern. That was just the first instance of a practice that's become incredibly commonplace. So we could all be in that scenario today and not know what our cells are being used for or in the ways in which they're being grown and turned into cell lines. This is another huge ethical territory that's being discussed so little, like most of these things. It's so little discussed. There was an opportunity a few years back, towards the end of the Obama administration, there was a reviewing of the regulations in the U.S. that guide the governance of discarded materials, that guide the governance of what we consider to be anonymous data or identifiable data. DNA data can never be considered anonymous. It can never be considered de-identified because you can always learn the identity of the person through their DNA. And that's only become easier over the years. There is tremendous potential for this enormously personal and identifiable data to be hacked, to be leaked. We're already seeing this happening with medical records so regularly that they're being hacked. And this is our most personal data, the DNA data that is the core of our biology. We should really give it a thought if we might want to protect it a bit more and involve people in understanding what is being done with it and give them some agency over what's being done with it.
You mentioned 23andMe, which I find a fascinating story because they actually pivoted into what they became known for, which was doing these ancestral DNA tests. And then recently they had financial difficulties, so they are now selling the company, and I assume all the data that comes with it.
Yeah.
It's what happens when you mix this DNA and identity with capitalism and commercialism?
Yeah, this is the situation that we're in. And again, we're somewhat at the beginning of it. But it's not as many people with their DNA profiles in 23andMe, for example, as in a lot of the social media platforms. But it's not so different. And what is to prevent these DNA companies from pursuing the same approaches that we've seen across social media that have been so damaging to the public fabric or to our ability to communicate, to our ability to relate to each other? So that's really the question, I think, is there's potential to build DNA social networks in a way that could be really about bringing people together, about understanding your connection to so many different people. And there was an aspect of that, I think, to 23. I found that when I logged in there and I said, oh, wow, I have 7,000 DNA relatives. This was amazing to think that you have connection to all of these different people that are really different. There is some opportunity there to push things in a way that can make people reflect on how interconnected we are. But it's really about where the focus is. And so often the focus has become quantifying things that have to do with ancestry. Like, what percentage European are you? What percentage African or Asian? This is unfortunately very divisive and seemed, at least for a while, did drive corporate profits and drove also talk shows and things like this, like figuring out what percentage you are really of this thing or that thing. And I fear that's just really the wrong direction to be going in. I hope that alternatives will come. I hope that there will be alternatives, that new companies will come, or maybe nonprofits. It doesn't have to be a company. Maybe that's the problem, maybe that was the problem with 23andMe, that there was never really a good plan for a good business model, right? This is not my terrain at all, but just as an observer, if this information was a public trust, if there was a public entity governing the use and interpretation of the data, it is really in the public good. So it raises this question about why it would need to be a profit-oriented business. I think that would be an interesting area to explore. I always think that with public media, why don't we have more public media versions of social media and spaces on the internet that are publicly funded? I would love to see more of that there as well. But also with biology, I think what we really need are these spaces or sites or entities where people are well consulted, where they have agency over their data, where they understand what's going on, where they go through a process of consent that is real, and where you have diverse stakeholders generating the information and interpreting the data and having discussions about what it means in a way that is contentious, that kind of tries to hack it out. Like, what does this actually mean? And have debates about it and so on. We need a platform to have that kind of activity happen where people can have trust that their data is being used in a way that they are consulted about, and that the meaning can be hashed out also in a way that is complex.
I'd be really interested to hear about your artistic practice and how you think about storytelling and shaping the narrative versus leaving open spaces for people to explore, because you've mentioned examples of both.
Yeah.
You've mentioned the books and the comic books, but also the opening of avenues and different opportunities through, for example, the many faces of Chelsea.
Yeah.
How do you decide how to hit the balance? Does it also depend on the question that you're trying to resolve or explore?
It definitely does depend on the content of the experiment. Each of my projects begins with some kind of question or something that I'm probing, trying to find out, like the hair example, right? How much can I learn about a stranger from a hair? You go through this experimental process and end up with all of these artifacts and thoughts, and you start thinking, how do I turn this into something that an audience can experience also? Sometimes that can be relatively straightforward, and sometimes it's really hard to figure out. Like when you're working with something like a virus, I worked on this project where we engineered a virus to insert an extra copy of the oxytocin gene into human cells. And so it was a love virus. How do you make that visible? It's really a challenge to think of, how do you make viruses visible to an audience? And so in the case of the virus, that led me to indeed developing this kind of speculative story, building these sculptures that would contain the virus, that would feature in this story as imagining that there would be this future society where people would break open the virus and consume it in these rituals together, and be more loving and intimate with each other and walk down the street holding hands with strangers. So that was a very narrative project. T3511, the film that I worked on, is also very much a narrative project. And then there's projects like Stranger Visions or probably Chelsea, where it's really these faces. You walk into the gallery and you see these 3D printed portraits and can draw a lot of different interpretations from that. And maybe you'll have on the wall a small label saying that these were made from DNA. And otherwise, it's left up to you. And so I think my preference is to say less. If I can find a way to say less and leave more space, more openness, so that people can come in and project the things that they're thinking about, the things that they're concerned about, also like through these materials, through these examinations of biology, genetics, biotechnology, that's definitely my preference. But if I can't find a way to do that in a simple way, then it gets more complicated. With Stranger Visions, it's like, okay, here's a portrait, here's a cigarette. The audience doesn't need me to tell them the story because they can easily conjure that up in their heads. And they conjure up all kinds of interesting stories from that. But with other projects, that story is not so obvious. And then I have to really spell it out in more detail. So I guess it's kind of like that. And maybe that's really also a question of how much my creative ability has led me to a place where I can step back. Because the most successful pieces are the ones where there's more space, more room. So that is what I aim for.
And since you spoke about some themes of what you're exploring at the moment, if we flip that and you mentioned wanting to create space for the people experiencing your work to think about things, what are the questions at the moment that you want to pose to other people?
Oh boy. Okay. So at the moment, I'm very absorbed at the moment in researching into genetic predictions of behavior. So this is the next phase of genetic profiling research, I guess you could say, is looking beyond what we might look like, beyond disease, to these attempts to predict people's personalities and behavior from DNA. And there is a substantial amount of research that's being done, whether it is predicting people's intellectual attainment in the future, or predicting sexual orientation, or predicting violence. So predicting violence, this is the one that I'm most focused on at the moment, working on a project that looks at, or is really working with an individual who's on death row, who had this idea of the violence gene, used in his sentencing. So he was sentenced to death in part because of the argumentation that he would be genetically predisposed to violence. Now, that is after having been found guilty of committing a violent murder. So it's not that he's innocent. This is not the point. But the idea of profiling a gene for something like a future violent behavior could already be entering into courts of law. This, I think, is pretty shocking to most people. And that isn't just restricted to the US. So that has happened in other countries around the world as well. So that's something that is very much occupying my attention right now. Oh, there's a whole nother thing that we didn't talk about at all. That's quite a bit lighter. Another project that it's always simmering. I'm working on this collaboration with the artist and psychoactive botanist, Philip Andrew Lewis. And we have this long-running project attempting to engineer plants, in particular psychoactive plants, to contain the DNA of loved ones. The idea is to have these new forms of mourning, of grieving, creating these new rituals that use genetic engineering. So we would take the DNA from your loved one and put it into the psychoactive plant, and then you could grow that plant. And when you came to the part of your mourning process where you felt ready to move to the next step, you could consume that plant and have this last journey of intimacy with that loved one. So that's another project I've been thinking about a lot recently. We've done different experiments, had some success in engineering different experimental plants with human DNA, and we want to take that to the next step and really build this memorial forest. That's what we've been working on for some years, to try to build support and make this happen. And it feels suddenly like there might be some movement that this might be becoming possible. So this would be the idea that your loved one's DNA could live on forever in a medicinal or spiritually meaningful plant within this special space that would be the genetic memorial forest.
It's very beautiful. Unfortunately, we are at the end of our time. It's been such a great conversation, but I will just ask you one last question that I would like to end on, which is, what makes you optimistic about the future?
Oh, yes. Yeah, there are so many negative impulses that we're seeing all the time. There's a couple things that make me optimistic. One is political and one is personal. So the very personal one is that I had a baby about 10 months ago, and he is just a source of total inspiration and hope. When you look at that little face and that little smile that's so full of love, and you just think anything is possible with this little creature. We come into the world this way, and there's so much potential there. You can't not be uplifted by that, I think. So that on the personal level. And then the other is that while there is so much discussion about the turn to, let's say, the right wing into more like hateful attitudes around the world in the young generation, it often glosses over the fact that there is also a huge opposition to that. When you look at young people, there is a tremendous amount of political engagement, and really there's as much engagement in doing things that are for humanity and for bringing people together, supporting people, being more humane. And I do still have hope that we will see the younger generations really move out of the kind of detritus that we've left. I think they're going to piece things together and come up with something that's like radically new and beautiful out of all of this mess that we've left.
Thank you, Heather Dewey-Hagborg, for revealing the beauty and unease hidden in our biological blueprint. Next week on Moonshot Radio, we speak with filmmaker and immersive media artist Gabo Aurora, who's using virtual reality to expand empathy and reconnect technology to what makes us human.